such a pleasure and a uh, privilege to welcome today's speaker, Ron Harris Warrick, whom I've known uh, for a very long time, for his entire Cornell career. And then he and I and his wife Becky known each other from before then. So Ron is a Cornell lifer, and to succeed at Cornell, you're expected to be great at two of three things: research first, teaching, and service. And Ron has been exemplary in all three. You can read for yourself from the auto from the blurb for this talk what he's done, but I'd like to say a few things from my own perch and observation. I mean, Ron has been a great colleague and my best friend for, like I said, forever. But Ron, as a researcher, is, is definitely was one of the leaders in his field of neuropharmacology, modulation, central pattern generators. For his entire career, he was funded by the NIH, a career of almost 45 years or more, right? Yeah, and Ron served on many NIH committees and councils, and so uh, he is, so he definitely checks all the research boxes, and you can read those. He's also had time to publish over 150 publications. His awards are incredibly numerous, recognizing his research, but I think what is known mainly on campus is that Ron is really one of our department, neurobiology and behavior, has always had a lot of really terrific teachers, and Ron is among the very best. Uh, and not just at stepping up when it's his term to do the teaching. He was very early on an advocate of interactive teaching, which started in the mid to late 2000s. And Ron's teaching awards are at least as numerous as his research awards. Let me just name some of them because you know me. So he won this SUNY Chancellor's Award for teaching. He won the Stinson Award for mentoring. He won the Stephen H. Weiss Presidential Fellow, and he was a Menschel, a Menschel Outstanding Teacher Fellow as well. So he is, and I was his chair for a while. He was my chair, and I can say that his courses were among in an esoteric subject, uh, neuropharmacology, which drugs in the brain, was one of the most popular courses in the department for every year that he taught it. And there are still students who want there to be a course. So you're about to get a real treat because, for my money, Ron is one of the best scientific expositors out there. So, Ron, over to you.
Well, thank you. It's a real pleasure to be here, and I hope I don't let you down after all those very kind things Ron has said. So I want to talk about psilocybin and more generally the family of psychedelic drugs, and talk to you about kind of two sides of them, the hippie side, the getting you high side, and then the possible medical applications of these drugs. Psilocybin, as probably many of you know, is derived from mushrooms, psilocybe mushrooms. It's actually found in other types of mushrooms, conocybe and paneoloas and others, which grow in Central America and elsewhere. In fact, some of these mushrooms grow in the northeast of the United States. Their psychoactive properties have been known for a long time. These drugs have been used for shamanistic and ritualistic purposes in Central America for thousands of years. There's archaeological evidence. The Aztecs used magic mushrooms, the psilocybin mushrooms. Cortez talked about it, and they referred to it as Teonanacatl, which means God's flesh. And so these drugs have a very, very long history. As I said, psilocybin is an example of the family of psychedelic drugs, which are some of the most potent mind-altering drugs known to man. They have dramatic changes in perception and dramatic changes in the way of thinking without making you smashed or crazy and so on. So as I said, I want to talk first of all about the psychedelic side of these drugs and a bit about how the brain actually does that, and then move over to new work about the psychedelic drugs as novel family of antidepressants. So what's the psychedelic experience like? Let me just say the psychedelic drugs include things like LSD, DMT, which is the active ingredient in ayahuasca, which some of you may have heard of, mescaline from the peyote button. All these drugs have the same actions in the brain. They have the same targets, they do the same things, and they evoke the same experiences. The differences between them mostly have to do with potency, how strong they are, and not that they have different things. So to give you kind of a feel for what the psychedelic experience is like, I'd like to read to you from the first description of an LSD trip. LSD was invented, it was synthesized by Albert Hofmann in 1943 in Switzerland, and he accidentally ingested some of this 25th example of the LSD family. And this is what he wrote about it. He said, I had great difficulty in speaking coherently, and my field of vision swayed before me and was distorted like the reflections in an amusement park mirror. You can see this in some of these. These are posters that were given for psychedelic rock concerts in San Francisco in the 1960s, and they try to do some of the things that the LSD experience and the psychedelic experience gives. The most outstanding symptoms were vertigo, visual disturbances. The faces of those around me appeared as grotesque colored masks, and obviously you see that here. Marked motoric unrest alternating with paralysis. I had a clear recognition of my condition, in which state I sometimes observed, in the manner of an independent neutral observer, that I shouted half insanely or babbled incoherent words. Occasionally, I felt as if I were outside my body. Everything seemed to sway, and the proportions were distorted like the reflections in the surface of moving water. With closed eyes, multi-hued, metamorphizing, fantastic images overwhelmed me. Especially noteworthy was the fact that sounds were transposed into visual sensations, so that from each tone, a comparable colored picture was evoked, changing in form and color kaleidoscopically. He's describing the phenomenon of synesthesia, where there's a mixing of the different sensory inputs, and some people are natural synesthetes. Olivier Messiaen, for example, a 20th century composer and organist, was a synesthete and used his synesthesia to help him write the music, thinking of the colors as he was doing it. So this is kind of, that was the first LSD trip, and many people since then have experienced these drugs are obviously very popular with hippies in the 60s. So to summarize the psychological component, there are perceptual distortions. We call these pseudo hallucinations because they're not true hallucinations like a schizophrenic experience where they can't tell that things aren't actually real. You retain insight so that you know that this is the effect of the drug. There are prolonged after images. This is another thing where if you move your hand quickly across your field, you can actually see the remnants of the hand for a little bit. And that's very much slowed down so that it almost feels like your hand is a long three-dimensional object, kind of slowly melting. If you've ever seen the Beatles movie, Yellow Submarine, which is all about psychedelic drugs. There's a lot of examples of these trailing phenomena in that. There's synesthesia, marked slowing of subjective time, and again, this retention of insight. With higher doses, you can sometimes get what we call the psychic phase, which is a pseudo mystical experience, which is characterized by a loosening of boundaries between the self and other, kind of dissolving in the universe, becoming one with the universe or becoming one with God or nature, depersonalization. And this is often accompanied by a sense that of great profundity, that you're having insights into the world and yourself that you never had before. And this is, psychiatrists call this portentousness. Let me give you an example of that from a book by Aldous Huxley, a famous writer who wrote Brave New World and Point Counterpoint. He also wrote Doors of Perception, which is a book about his experiences with mescaline. And this is what he said. So he was sitting in a comfortable room in his home with a person he was talking to. And along with indifference to space, there went an even more complete indifference to time. There seems to be plenty of it, was all I could answer when the investigator asked me to say what I felt about time. The investigator then told him to look around the room, and he saw a wicker chair. And something very, very portentous happened with this experience of the wicker chair. He said, I had what I can only describe as the sacramental vision of reality. I was in a world where everything shone with the inner light, that's capital I, capital L, inner light, and was infinite in its significance. The legs, for example, of that chair, how miraculous their tubularity, how supernatural their polished smoothness. I spent several minutes, or was it several centuries, not merely gazing at these bamboo legs, but actually being them, or rather, being myself in them, or to be still more accurate, for I was not involved in the case, nor in a certain sense were they, being my not self in the not self, which was the chair. So this is an example of profound thinking and changing of your attitude towards things around you. Uh, Sometimes they are very profound and sometimes perhaps not quite so profound. And I'd put that in the not quite so profound state. One other thing about psychedelic drugs that sets them apart from all other psychoactive drugs is that the experience depends entirely on what we call the set and setting. If you get drunk or take cocaine or heroin, you're going to have the same experience pretty much no matter where you are. That's not true with psychedelic drugs. So the set is the mindset that you bring to it, what you think is going to happen, what you expect. For example, the Native American church, the largest church for Native Americans in North America, uses peyote, uses mescaline from the peyote cactus, the peyote buttons, as a sacrament. And they use that to experience God. And they expect the setting for them is they're going to have a religious experience and interact with God. And this happens. Then the setting is the situation in which you take it. For the Native American church, they do it in a religious setting with lots of religious symbols. And the idea is that they will have a religious experience, and they do. Quite different if you take it at a rock and roll concert or a rave or something where there's no control, it's kind of scary. And in those situations, people have the famous bad trips where they think they're going crazy and so on. These typically only last a minute or two. They're not lasting or of long duration, but they can be very frightening. Let me finally say these drugs are not addictive. They don't activate the addiction pathways involving dopamine. And tremendous tolerance builds up. You can only take the drug for a day. If you try to take it the second day, you don't get any effect. And it takes up to a week to restore your sensitivity to these drugs. So no one gets addicted to them. So that's kind of the experience. And I'm a neurobiologist, so obviously very interested in trying to understand how it's working. I've lost my, oh, there it is, okay. So now we're trying to think about the brain. So let me give you a little bit of basic neurobiology. And if you're not interested in this, you can just tune out for the next couple of minutes. So the brain has 100 billion nerve cells, and each one receives about 10,000 inputs from other neurons. And the inputs where one neuron talks to another neuron are called synapses. And these are various pictures. It's an electron scanning electron micrograph of the synapses, where all these little red and yellow dots are synapses where one neuron is talking to another neuron. Let me just briefly say that the neuron has many processes sent out that are called dendrites, and that's where most of the synaptic inputs are located. And in many cells, these dendrites are covered with spines, these little spines that are coming out here. That's where the excitatory synapses are made, and I'm going to come back to these spines later in the talk. If we zoom in and look at a synapse, this is it. And basically what happens is that there's an electrical signal generated in the cell. And when it gets to the end of the cell, that electrical signal triggers the release of a chemical substance, a neurotransmitter, which then is released from the terminus and diffuses across the space to the next cell, where it interacts with a very specialized protein called a receptor that recognizes that molecule and only very closely shaped molecules. So there are receptors for dopamine, and there are receptors for norepinephrine, there are receptors for acetylcholine. And then when they bind to the receptor, they activate the receptor and cause a response. And eventually they fall off the receptor, bounce around a bit like pinballs, and then get caught and recycled by a transporter that transports the transmitter back into the presynaptic terminal where it can be reused. So this is kind of the battleground of psychoactive drugs. Virtually all the psychoactive drugs act at synapses to alter the function of synapses. And the psychedelic drugs do that too. A hint of what they might be doing comes from their structures. So again, the details here are not interesting, but they look a lot, many of them look a lot like a neurotransmitter called serotonin. So this is serotonin, 5-hydroxytryptamine. And if you just look at the blue part, you can see the structure is repeated. Here's psilocybin, for example, and it has the same structure. This is DMT. This is the active ingredient in ayahuasca, has the same structure. Here's LSD, where it's this kind of a sterically constricted form of the same shape. So the idea that they look like serotonin leads to the idea that they might actually act on serotonin. So serotonin is a transmitter that mainly functions to kind of set the gain of the entire brain. It's not involved in point-to-point, say, perception or activation of locomotion or something like that. It's really kind of changing the excitability of the brain, and plays a role in lots of things. You've probably all heard of SSRIs for the treatment of depression. These are serotonin selective reuptake inhibitors, and what they do is block this transporter, therefore preventing the serotonin from going back into the cell and just having it allowed just to keep bouncing around and keep reactivating receptors. So you get an exaggerated serotonin response. Serotonin is also involved in sleep and dreaming. For example, the only time the serotonin cells aren't active is when you're in deep sleep. All the rest of the time that you're awake, they're active in releasing serotonin. Plays major roles in migraine, appetite control, pain perception, motor coordination, impulsive aggression, and so on. So the fact that these drugs are shaped like serotonin leads to the idea that psychedelics might actually act by, in some way, interacting with the receptors that serotonin interacts with. And that turns out to be correct. Here's a list. There's a whole lot of different types of serotonin receptors, each of which does something different. And LSD and the psychedelic drugs interact with almost all of them, as you can see here. It turns out that the entire psychedelic effect is due to interaction with one of these receptors called a 5-HT2A. So let me show you a graph. I hope I'm not scaring you. This is a graph that compares the potency of the psychedelics to its ability to bind to that receptor. So on the x-axis, this is the dose, how much you have to take in order to get high. And on the y-axis is how well it binds to the receptor, how tightly it binds to the receptor. And this is a complicated slide. It's a log plot of the inverse of those things. But what you really see is this straight line. That is, drugs that bind very tightly to the receptor, like LSD, work at very, very low levels, microgram levels of LSD. And drugs that bind very poorly to this 5-HT2A receptor, like mescaline, take many milligrams and possibly grams to have an effect. Psilocybin is kind of in the middle. It takes 10 to 30 milligrams to have an effect. Other evidence is that you can do your fancy genetic engineering nowadays and build mice that don't have a 5-HT2A receptor. And mice actually respond to psychedelic drugs with changes in gene expression and a very funny kind of a head nodding behavior. And these effects are lost if you get rid of the 5-HT2A receptor. And there are drugs you can give to humans that temporarily block those 5-HT receptors. And they don't have any psychedelic response. So the receptors are there, they're on a subset of the cells in the brain. Serotonin's kind of bathing the brain pretty much whenever you're conscious. So why aren't we high all the time? You know, there's receptors, there's serotonin, these things are acting like serotonin, but we aren't high all the time. And this is because these drugs do something really amazing. They hijack that receptor and make it do something it doesn't normally do. And this is a phenomenon known as biased agonism, where a receptor it's a protein and it can change its shape in different ways. And so when serotonin binds to it, it changes its shape so that it triggers a set of biochemical events inside the cell, the first step of which is binding to an internal protein called a G protein. There's a whole lot of different kinds of G proteins, and each one of them activates a different biochemical pathway and causes something different to happen. So when serotonin does it, it's actually GQ. The psychedelic drugs can do this, but in addition, they can, when they bind, they cause the protein to change its shape so that it no longer binds GQ, it binds a different G protein, GI, that never happens with serotonin. So this is an effect that you can't get with serotonin itself. You only get it with the psychedelic drugs, and all psychedelic drugs do this, and no non-psychedelic drugs do this. So this is basically how it is happening at a molecular level. And for anyone that wants details, I'm going to skip over that. So how does this get you high? Well, we don't really know, but we know a couple of things. These drugs cause a tremendous release of the major excitatory neurotransmitter in the brain called glutamate. And that makes many, many, many cells active. That you can measure, this is measuring glucose metabolism in a human brain. And the green areas are areas that are hyperactivated during psilocybin. They include prefrontal cortex, which is kind of where our normal thinking and rational analysis goes on. The anterior cingulate cortex, which involves emotional and internal representations. The visual cortex, that probably has something to do with the hallucinations. And very interestingly, this area down here, it's called the thalamus. It's kind of the gateway for sensory inputs to the cortex. Almost all of our sensory feelings, vision, hearing, touch, and so on, funnel through different nuclei in the thalamus, which then sorts them out and sends them to different parts of the cortex where we become aware of those. And this is kind of the gateway to the brain for sensation. A very recent paper has looked at what's called functional connectivity in the brain, where this is using functional magnetic resonance imaging. And you can see that there's certain parts of the brain that correlate in their activity and kind of go together. And these can form networks that can be mapped and that are activated under different circumstances. What happens with psilocybin is all these networks get confused and mixed up and desynchronized, okay? They're all weakened, and this is something called Euclidean distance between these networks, and the bright red that you see here is showing a reduction in the strength of all this. So the normal systems that we use to interpret the world are all mixed up, okay? That reduces, for example, the thalamic filtering of sensory inputs so they can get confused and you start having effects like synesthesia. Decreases integrated processing so that you make connections between you and a chair that you would never make under normal conditions. And interestingly, this correlates with the strength of the mystical experience. One thing the psychiatrist asks is how much you feel oceanic boundlessness. So this is a checklist. Hey, are you experiencing oceanic boundlessness? And if you do, the strongly you do that, the more disorganized and desynchronized your brain is. So that's kind of the end of the psychedelic side. Let's switch over to the antidepressant side. So we all either have been depressed or know someone depressed, and we know the difference between being sad when something bad happens and being clinically depressed when you feel like you're at the bottom of a black hole and don't see any obvious way out or any way forward. And this is a terrible, terrible disease, the most common major psychiatric disorder in humans. The DSM-5, the Diagnostic and Statistical Manual of Mental Disorders, has a list of criteria, which I won't go through in detail. You have to have continuing sense of depression for at least 2 weeks, and 4 other signs, the most common of which are anhedonia, which is a failure to find pleasure in normally pleasurable things, your favorite food, a good wine, sex, whatever. Very often are feelings of worthlessness or of guilt, and there are a number of other signs, among which is recurrent thoughts of death and suicide. 3/4 of the suicides in the world are caused by clinical depression. Untreated, this can last a long time. Most people get drugs now, at least in the first world, but it can last months to years. It's more often than not a cyclic disease that comes and goes over time. This is major depressive disorder, and starts young, late teens and 20s is when people start having their first experience. Our students, many of them are having a problem, because this is a very common disease that affects 1 out of 5 people in the United States will have a clinical case of depression during their lifetime. Interestingly, it's about twice as common in women than in men, for a number of reasons. If you're interested, we can talk about that later. So we need some new antidepressants. Basically, there haven't been any new families of antidepressants for many, many years, over a decade. And we keep using the same ones. The SSRIs like Prozac and Celexa and so on, the SNRIs, and the SSRIs, remember, block that serotonin transporter. SNRIs block serotonin and norepinephrine transporters. NRIs block norepinephrine transporters, and then there's a bunch of others, all of which are kind of doing the same thing. And they don't work very well. Basically, they're only effective in about 2/3 of patients and not fully effective in even half of those. So there's 30 to 40% of people with depression that are called treatment-resistant depression, where none of the current drugs help them. The other bad thing about current drugs is they take a very, very long time to have an effect, weeks to months. And this is terrible, because here you are depressed, you say, I've done all the right things, I've gone to the psychiatrist, I've got my prescription, and I'm taking the drugs, and I'm not feeling better. And a week goes by, and I'm still not feeling better, and 2 weeks go by, I'm still not feeling better. And thoughts of suicide come in, and many, many times people commit suicide during that time when they've already started the antidepressants, but they haven't kicked in, they haven't had an effect yet. So if we could find drugs that would act rapidly, we could save a lot of lives, right? But we need them to act within days rather than weeks or months. Now, one recent one is currently available. It's called ketamine. Ketamine is a dissociative anesthetic, and its major use up until now has been for veterinary surgeries. I use it myself in my lab when I do surgeries on mice. I give them ketamine and xylazine. It's a dissociative anesthetic, so it was also a party drug. It's been used in raves and so on, because you have an out-of-body experience and a lot of distortion of visual things. But it's also a very rapidly acting antidepressant. Within hours of taking ketamine, depressed people feel better. And they feel a lot better Unfortunately, it doesn't last very long, a couple of days, maximum of a week. So this is not a panacea, but it has now been approved by the FDA for treatment-resistant depression, the ones that the standard drugs don't work. And there are people experimenting with giving ketamine during that months when they're also taking an SSRI, but the SSRI hasn't kicked in yet, and the ketamine taken once a week can kind of keep you going and non-suicidal during the time that the drug kicks in. So this is not as what we'd like is a drug that has a longer fast onset and long action. Now, are the psychedelic drugs those drugs? It's an open question, but let me show you some of the data. So this is about psilocybin. This is an early experiment done in 2021 where basically a group of people were broken into a control or a psilocybin group. They were given some therapy beforehand. They were given two trips with psilocybin in the presence of a therapist, where the set and setting were very positive for these people. Comfortable room, nice music, whatever. And then they asked how their depression was using a scale called the Hamilton Depression Scale, which rates you on a number of features where a value of 7 or less means you're not depressed. And then there's moderate, and there's mild, moderate, and severe depression. Severe depression is 24 and above. So they took people with moderate to severe depression and gave them this experience. And what you see 5 weeks later is a very, very significant drop in the level of depression. The average level of depression was 8, which is just the slightest hint of a clinical depression. And week 8, 3 weeks later, it was still that low, okay? And many people were 0. They had completely gotten rid of their depression. So another paper followed up in 2022 that carried this out further. Again, this is the Hamilton scale. And they found that even 1 week later, the depression had already stopped, and more recent data has now shown that there's significant drop in depression within 24 hours, and then it continues to grow, where it kind of bottoms out at about a week. And this carried the study out to a year, where you see that the reduction in depression for many of the people, not all of them, but for many of the people was very long lasting. This is after 2 treatments with the drug, no drugs after that, no side effects of drugs after that, because the SSRIs have lots of terrible side effects. This is just no treatment, and the depression remains down. So this is hopeful. And based on these kinds of preliminary experiments, the FDA approved what's called breakthrough therapy that allows accelerated trials of this. And it's been through phase 1 and phase 2 trials. Phase 3 trials are the really big trials with hundreds and thousands of people. And those trials are ongoing now. So we don't know whether this is actually all going to pan out. People are very hopeful. We should have the answer within a year or 2, I would guess. So I asked, well, how does psilocybin do that? You know, how can it be an antidepressant, psychedelic drug? You know, so here's 3 possibilities. The first one is it's the mystical experience. The mystical experience completely changes your relationship to the reality of the world around you, to the people you're with, in such a way that you've had kind of an aha moment, and your depression is affected by that. A second possibility is that psilocybin enhances the psychotherapeutic interaction with therapists so that it's much more efficient and effective than it normally is. And the third is a biological action. It rebuilds the brain via the 5-HT2A receptor or some other serotonin receptor. Now, these are not mutually exclusive. All 3 could be true. It's interesting with regard to mystical experience that people remember that had the strongest mystical experience had the greatest disorganization of their brains with those bright red brains I showed you. They also had the strongest antidepressant response. So there's quite likely something going on there. With regard to suggestibility, a very recent paper suggests that may not be quite so important. They did a meta-analysis of a bunch of studies where psilocybin was given under different conditions where in one set, the therapist was acting like a therapist and was guiding people through. Some of them even had manuals that would lead them through. And in others, the therapist was just there and gave them water when they wanted water and kind of acted supportive but didn't do anything. And it turned out the antidepressant effect was the same in both cases, in all these cases, suggesting that it isn't an increased suggestibility, but that's not proven yet. All of these things, of course, affect the brain. The brain is what we think with. So a biological effect could be true. So there's reasons to think that's true. And for one thing, psilocybin is a good antidepressant for animals who don't have psychotherapists and presumably don't have much of a aha moment of mystical perception. Mice, for example. Depression is a human disease, so you have to believe in these models of depression in animals, which show different types of validities. A face validity means it looks like depression. Pharmacological validity means antidepressants block it. And there's a bunch of ones. The most popular one is called continuous intermittent hassling, I can't remember the word, where basically you put a mouse in a cage and you do bad things to it over a period of weeks. One day, a bright strobe light will go on randomly. The next day, a loud horn will go on randomly. The third day, the bedding is wet. The fourth day, there's no food. The fifth day, you put a much larger mouse in, which chases the little mouse all around. And you keep doing this every day for weeks. And after 3 weeks, the mouse is huddling in a corner. It's avoiding all other mice. It has anhedonia. It will not have sex with the opposite sex. It will not prefer sugar water over regular water. And it looks depressed. And furthermore, antidepressant drugs like SSRIs will relieve that depression. The animals come back to normal. And so does psilocybin. Psilocybin works just fine, and it works much more rapidly. For the SSRIs, it takes weeks for the animals to get better. With psilocybin, it's the next day. So the idea is that psilocybin is actually doing something on the brain. And we have an expert here at Cornell in the Department of Biomedical Engineering, Alex Kwan, who's actually working on this. And he's made a hypothesis that what psilocybin does is to enhance synaptic plasticity, these connections between the brains. And the idea is that depression itself is associated with a loss of synapses at certain places in the brain, in the cortex, a diminution of the number of connections, and that the antidepressant effect is a restoration, building new synapses to replace the ones that were lost. And they've coined a word, psychoplastogen, for drugs that affect the synapse formation. So there's some evidence for this idea. So the idea is that these drugs cause this massive release of glutamate, which then highly excites the dendrites, the recipient parts of the neurons, causes a big increase in calcium, which is the second messenger that triggers all sorts of biochemical events, which eventually result in new synapses being formed in the parts of the brain where there are 5-HT2A receptors. So here's some evidence. Here's an animal study in mice where basically they're looking at neurons in the cortex. These are called pyramidal neurons, and they're covered with these spines that I mentioned. These are the sites where the excitatory synapses are formed. And then they put the animal through this depression system, and what you find is that there are far fewer spines on the dendrites of these animals. And then you give them psilocybin, the spines come back. And they actually come back pretty much the way they were before. What Alex is doing now is doing this in vivo. He can actually look inside the brain of a living mouse. And when the mouse is depressed, there aren't very many spines. And here, his lab tried 3 different psychedelics, LSD, DMT, and DOIs, an artificial compound. All of them cause this increase in spine formation within 1 to 3 days of when the drug is given. This is one single treatment. Now, humans, we're not so sure. We can't look into the brains of humans. There have been post-mortem studies in humans showing a loss of spines in depressed humans who never received treatments. These are mostly suicides. So that's a confounding problem. So what people are doing is looking for something that'll tell them how many synapses there are. And what they're now using is positron emission tomography, where you add a radioactive tag to a molecule that specifically binds to a single protein and then puts out its signal, and you can measure where that protein is and how much of it there is. So they've done this for a protein called SV2, which is in synapses. So if there are more synapses, there will be more binding to SV2 and you get a brighter signal. And they did this for normal people, people with mild depression, and people with severe depression with Hamilton scores of above 20. And the brighter the color is, the more synapses there are in these human brains. And what you see with mild depression, it's pretty much the same as the control, but with severe depression, there's a significant decrease in the number of synapses that you can see this in the dorsolateral prefrontal cortex, the cingulate cortex, hippocampus, which is involved in learning and memory. Interestingly, if you give these people psilocybin within a couple of days, that reverses. Okay, so here's the hippocampus. Within 1 day, the number of synapses goes back up. And at a week, it's even stronger. The prefrontal cortex, a little slower, takes about a week to have a big effect, the increase in number of synapses. So the idea then is that depression has many components, that it's not just loss of synapses, but loss of synapses is an important component. And if you reverse that, you can get rapid relief from depression. So two final questions. We know that the psychedelic effects of these drugs are mediated by binding to 5-HT2A receptors. The antidepressant effect doesn't have to be mediated by that receptor. It can do something else. Remember, these drugs bind to many different types of serotonin receptors. And so it might be possible to design some drugs that are antidepressant, but don't bind to the 5-HT2A receptor, in which case it might be possible to have new drugs which are rapidly antidepressant and not psychedelic. I mean, some people may look forward to psychedelic experience, you know. The people that were in the clinical trials for depression, 2/3 of them rated their experience with psilocybin as one of the most important events in their entire lives, one of the 5 most important events in their lives. Most people don't want to do that, and with current treatments, you have to take it with a therapist who's there to kind of help you, support you, and make sure that the setting are correct. If we could come up with drugs that weren't psychedelic, then you could just take it at home and, you know, twice, and then you'll feel better for maybe a year or more before you have to do it again. The drug companies are all over this. There's a lot of money here. And so they're starting to come up with a large number of drugs which appear to do just that in animals. No human studies have been done. So DMT is very closely related to psilocybin. These are some variants on DMT. There are some drugs that we know, in fact, interact with 5-HT2A, but are not psychedelic, like 2-bromo-LSD. Ibogaine is a drug from the iboga tree that's found in Central Africa, a psychedelic drug, so they're modifying that. There are modifications of mescaline, the active ingredient, peyote buttons, and this is the part of that biochemical pathway I skipped over. So this has only been tested in animals, but the general take-home message here is that we're kind of in the middle here. It's not fully worked out, and we're not sure that this is working, but it's very hopeful, because for decades we've been turning the same wheel. It's all serotonin reuptake inhibitors or norepinephrine reuptake inhibitors or things that affect that. This is an entirely new direction. And it's a direction that has promise of acting rapidly and acting for a long time with only brief treatment. Okay? So this is not the only thing these drugs might be useful for. Even back in the 1960s, there were psychiatrists looking at the many applications of this, and there are currently clinical trials going on for anxiety, post-traumatic stress disorder, and various types of addiction, alcoholism, smoking, heroin addiction. So this may, right now it's all promise. It looks good. The preliminary data are all very good, but this could really open new windows to our treatment of one of the most devastating illnesses in humans, of depression. So thanks for your time. Any questions?
We do have time for questions. We have one on Zoom so far. It says, as we know, the frontal lobe of humans is not fully developed until around the age of 25. How do these studies apply or not to that population?
That's certainly true. It takes a long time for the brain both to get fully myelinated, and actually one of the major things that occurs over that time is synaptic pruning, the simplification of the connections. When the brain's first set up, there are far more synapses than a couple of years later. Depression has its typical onset in the early 20s. There is depression in very young children. It's rare. And no one's ever done any studies of treatment of young children with psychedelic drugs yet, so I don't know the answer to that. There are also, of course, changes with aging, and depression is less common in the aged than it is in the kids in the 20s, but it still occurs. Some people have first episode of depression over the age of 60, and there are continuing changes in brain structure and brain function during that time. So they're asking a really, really interesting question that does this final maturation of the brain in some way lead to the increased probability of depression at that time, and whether these drugs will selectively help that age? This work just hasn't been done yet. Good question.
My understanding is that.
Yeah.
I was trying to talk loud.
Sorry, I'm hard of hearing, and I hope I'm not the only one in the room that's hard of hearing.
Thank you. My understanding is in these various clinical trials of psilocybin, the people over a certain age, I'm not sure what that age is, are not included in those studies. Is that correct, and why?
Yeah, that's partially correct. Actually, the very first studies of the antidepressant effects of psilocybin were done with the agent. They're done with people that had terminal cancer and were dying and were suffering depression as a consequence of that. That was the actual first study that was published. And it was very, very, very effective on those people too. 70–80% reported a relief of their symptoms. Clinical trials are always a problem. The phase 1 and phase 2, they usually do limit in order to try to get more homogeneity. Phase 3 trials cannot do that. They must be much, much broader. So the phase 3 trials will definitely include the agent in it.
Hi. So SNRIs are often used for like chronic pain management.
Sorry, just start again.
Oh, do I have to hold something? Oh, SNRIs are often used for chronic pain management. So do these class of drugs, do they show any promise for chronic pain treatment?
Yeah, you might be thinking of TMS instead of fMRI. fMRI is a way to visualize blood flow in the brain. And TMS, transient magnetic stimulation, is a way to apply magnetic pulses that then can reconfigure the cortex and know deeper.
I was thinking the serotonin.
Oh, SSRIs, yes. Yeah, there haven't been any studies of these drugs for pain, but SSRIs have been tried for lots of things, and they work better or worse on others. I haven't heard much about SSRIs in pain.
I wonder if you know anything about these drugs helping people in our age group, which you say hasn't been tested very much, with cognitive decline or Alzheimer's.
There have been a few studies on that, and they don't seem very impressive. So it doesn't seem to be working for Alzheimer's. But it hasn't been studied in great detail, but the preliminary results are not promising.
I used to teach a course called Sacred Dimensions of Healing.
Great.
And we used a book by a longtime Johns Hopkins researcher I think his last name was Richards, and it was called Sacred Knowledge.
Okay.
You know that? Okay. Anyway, so he's describing many years of using psilocybin in treatment for people with depression. And I think I would perhaps say that if you developed a drug that didn't have the psychedelic properties, it might well relieve depression in people, but it would not give them the profound experience that many of them had, wherein they lost their fear of death, for instance, and then lived on in a state of greater tranquility and even joy than they had been able to before. And I think that's because the psychedelic drugs challenge your basic feeling about reality and about yourself. I mean, we know that, basically we adopt a hallucination as reality, that our brains are making a picture of reality, based on what our senses give to us, but it's a particularly human version of reality, and it's common amongst us, so we can say that we all consent to it, a consensual reality or something like that, but it's not reality as such. And we have an idea about ourselves too, which is also a construction that our brain makes. The psychedelic experience challenges both of those, so that, well, if I could, get personal here for a moment. I, of course, experimented with psilocybin back in the 1970s, and then I left it alone until about 2020, when I returned to it. And for the last 5 years or so, I've been doing it once or twice again during the summer usually, and usually at our lake cottage, Cranberry Lake. And what happens for me under the influence of this is that I seem to perceive subjectively something that I know intellectually, but couldn't perceive otherwise. So for instance, I'm looking out at the area in front of me, which then leads down to a lake, bathed in sunlight. We can't, of course, see light. We see its reflection.
Right.
We can feel its heat if we're out in it, but not if you're in the shade.
Yeah.
But under the influence of psilocybin, I really felt the energy of the sun coming down, bathing the scene in front of me,
So
and
I
all the
think-
green leaves and blades of grass vibrating with that energy.
Right.
You know?
So I basically agree with you. I think it can have extraordinarily profound effects. This is why Aldous Huxley called his book Doors of Perception. What he was referring to is the effect of mescaline to open the doors of perception and to cut off the filtering that makes each of us have our own person defined and societally defined interpretation of what reality is. By opening those doors wide open, he was arguing that you saw the real world for the first time. The fact is, there's a lot of people that just don't want to have a psychedelic experience, and many of them are depressed. And you can either forget about those people, or you can try to design drugs that aren't psychedelic. The psychedelic drugs, hopefully, I mean, who knows with the new FDA what's going to happen? Probably less likely than it was a year ago that these drugs will be approved. But they would be available for people who want to have the advantages that you talked about, and for people that don't want them, I think we can't ignore them. We have to make a try.
I have two questions. One is do panic attacks have some relationship to these topics? And the other is whether really
Can we do it
advanced.
one at a time, because I can
Okay,
never
yeah,
remember
okay,
two questions.
Okay, panic attacks.
So panic attacks are when you have this depersonalization, this effect, your kind of personal ego is dissolving. This can be very frightening. You say, am I ever going to come back? What am I going to tell my girlfriend? And well, I've become one with the universe, and so our relationship's over. And it can make people very, very frightened. In the appropriate setting, this almost never happens. It's very, very, very rare. It was more common in the 60s, and there's a lot of stuff in the newspapers about panic attacks and bad trips. This is because people were taking them without the appropriate setting. So that problem has been, to a significant extent, dealt with now. Now the second question.
So, well, this is a follow-up to the first. So when people have panic attacks, apart from any drug experience,
Mhm.
is that related to any of this in a way that you would
No.
address? Oh,
So panic attacks, there's a whole chapter in the DSM-5 on anxiety disorders. And the worst is generalized anxiety disorder, which, by the way, has very, very strong concordance with major depressive disorder. Many people have both major depressive disorder and generalized anxiety disorder. And panic attacks is a subpart of that chapter. It's a different thing. And I think the kind of panic attacks that those people have is really unrelated to the panic attacks with psychedelics.
so question 2 is whether you think really successful advanced meditators somehow enter into this equation.
Personally, yes, I do. And I think that's one of the real benefits of meditating. I've done a lot of meditating in the past in my life, and it was always very, very, very good for me. And there've been a lot of studies of brain function in meditating monks, which show some of these deregulations and so on that I was talking. So I think that is a very good direction and a non-pharmacological one.
Earlier, you observed that women are more prone to depression than men. Would you elaborate on that, please?
Yeah, so this has been known for a long time. It's almost a twofold difference. And there's a lot of, it's still not worked out. The current prevailing hypothesis, it has something to do with the cycling hormonal levels associated with the period in women. There's rare depression in young children, but up until the age of puberty, it's equal among boys and girls. And after menopause, this 2-to-1 difference disappears also, and it becomes the same. The onset of depression and the depth of depression is often triggered or is greatest in the premenstrual period. And so current thoughts say that it has something to do with estrogen and sex hormones and so on. There's also a couple of papers suggesting that testosterone may actually give some kind of protective effect. So it's not merely that women have cycling estrogen levels and men don't, but that men have higher levels of testosterone than women. This is not worked out yet, but that's the current hypothesis.
We have time for one more question.
Fascinating research, and thank you. Treatment is currently illegal in New York, correct? I know there's some
Yes. It's all in
states
clinical
and
trials.
cities where it's been legalized, but
Oregon, it's been legalized. Yeah.
so this treatment is not available legally at this point. In this
Except for
area.
Oregon, yeah. I mean, people go to Mexico as well. And South America, ayahuasca is kind of hot among movie stars for some reason. But what it requires is successful completion of the phase 3 trials. You know, these early studies all look great. They look fantastic, but until you have a large trial with a large population, a more diverse population, you can't really be sure that it's going to be good for the general public and therefore generally available. My guess is that when it is made legal, it will be made legal with a requirement that it be taken under supervision with a therapist. That's currently true with esketamine. You can't take ketamine on your own because you get high, but you can take it with a therapist. Fortunately, with esketamine, it only lasts a couple of hours, so it's not a long period of time. Psilocybin lasts 4 to 6 hours, so it's most of the day. But that's what I would guess it'll come out, unless we can come up with a non-psychedelic version, and then that might become generally available. Okay,
Let's
well,
give
thanks very
Ron,
much.
let's give Ron a great hand.